Thanks for joining us on another exciting journey into the world of science right here on the SANS Science Experience. If you enjoy this kind of content, hit like and subscribe. It's the best way to support the channel and keep these episodes coming. Today we're embarking on a deep dive into, well, a really fundamental question in biology. How is the process of growing up connected to how long we actually live? And we're going to explore this using a tiny but a really powerful model organism, the fruit fly, Drosophila melanogaster.
That's right. We're focusing today on a specific research article, a really fascinating one, titled NF-Mediated Developmental Delay Extends Lifespan in Drosophila by Kang and colleagues. And it really gets into a puzzle that's been around in the aging field for quite a while.
Absolutely. So our mission, you could say, is to unpack how these steps a fruit fly takes, from larva to adult, how those developmental processes can have such a profound lasting effect on its adult lifespan. We'll be looking closely at two main players here, a hormone called Ptth and a key part of the immune system, this thing called NF-BB signaling.
And what's really neat about this study, what makes it stand out, is how they managed to separate the timing of development from the rate of growth. That's often been a confounding factor in longevity research.
Right. Separating those two has been tricky. Okay, so let's set the stage. What's the basic puzzle they're tackling here?
Well, it's this observation we see pretty widely in the animal kingdom. Generally speaking, individuals or species that take longer to mature, they often live longer too.
You see that with larger animals, right? Longer development, longer lives.
Exactly. Elephants versus mice, for instance. But the big question mark has always been the why. What are the specific genes, the mechanisms linking that developmental timeline to the actual lifespan?
And that's where it gets murky, because so many genes that affect how long you live also affect how fast you grow, right?
Precisely. It's been hard to tease apart whether it's, the slower growth itself or just this stretched out developmental schedule that really matters for longevity.
So how did the study approach it differently?
They used Drosophila, the fruit fly, and focused on this hormone, Ptth, Prothoracicotropic hormone. Bit of a mouthful.
Well, yeah, Ptth. Got it.
So Ptth is basically the master timer for development in flies. And what they found, and this is clever, is that if you reduce Ptth activity, the flies take longer to develop. But crucially, their rate of growth doesn't really change much.
Okay, wait. So they develop slower, but they keep growing at a normal rate during that longer time, meaning they end up bigger.
Exactly. They end up as larger flies because they had more time in that larval growth phase.
Which is counterintuitive for flies, isn't it? Usually bigger flies are thought to live shorter lives.
That's the common wisdom, yes.
Yeah.
But here's the twist. These larger flies, the ones with delayed development from low Ptth, they didn't just get big, they actually lived significantly longer.
Wow.
And not only that, they were also much better at handling stress, like starvation or heat.
Okay, so that really points towards the timing itself being important, separate from the growth rate effect we usually think about.
It strongly suggests that, yes, the duration of development seems to be a key factor programming their adult lifespan.
Fascinating. And they dug deeper into the mechanism, mentioning ecdysone signaling. What's ecdysone's role here?
Right, ecdysone. Think of it as the go signal for molting and metamorphosis in insects. It's absolutely critical for moving through development. Ptth essentially controls the timing of ecdysone production.
Ah, okay.
And the study found that if they gave ecdysone back to those Ptth mutant larvae, the slow developing ones...
Let me guess, it sped up their development.
It did. It rescued the developmental delay. But, and this is the clincher, it also completely wiped out their lifespan extension.
No way. So the longer life was dependent on having lower ecdysone signaling during that extended development.
It seems so. The reduced ecdysone signaling, likely resulting from the lower Ptth, appears to be a key part of why they live longer.
And there was another piece, too, about inflammation. Inflammaging.
Yes, inflammaging, that low-grade chronic inflammation that tends to increase with age. They looked at the fly's Oenocytes.
Oenocytes, those are like the fly's liver cells, right? Metabolic hubs.
Pretty much, yeah. Involved in metabolism detoxification, and they found that in these long-lived Ptth mutant flies, the Oenocytes showed less of this age-related inflammation.
Okay, so the picture is getting clearer. Low Ptth leads to slow development, probably via low ecdysone, which results in less inflammation in key tissues and ultimately a longer life.
That's the emerging picture.
Yeah.
But to really nail down the molecular link, they turned their attention to NFP signaling.
NF-kB, we hear about that a lot with immunity and inflammation in adults. What's it doing during development?
Well, that's the thing. Its role in adult immunity is well known, but how it functions during development is much less understood. This study found that NF-key signaling actually ramps up during that critical larva-to-adult transition in flies.
Okay.
And it looks like Ptth, acting through Ecdysone, is what induces this developmental NF-key activation.
So Ptth exosome NF-key activation during development, how does that connect back to the longer lifespan of the Ptth mutant?
This is where one of the coolest experiments comes in. It's a real aha moment. They used a genetic trick to temporarily silence Relish. It's a key NF-key protein in flies, but only in the Oenocytes and only during an early larval stage.
Very specific targeting. And what happened?
Get this. Silencing Relish just in those cells, just at that early time point. It delayed development, just like the Ptth mutants.
Yeah.
And it significantly extended their adult lifespan.
Wow. So directly manipulating that immune signal in those specific cells during development changed how long they lived later on.
Exactly. It's a powerful piece of evidence linking developmental immune signaling directly to longevity. It suggests these early signals have really long-lasting effects.
That's quite something. It's like setting a program early on.
It really seems that way. And they also found that the Ptth mutant flies themselves, the naturally long-lived ones, showed less immune activation as they got older. Things like antimicrobial peptides and the activation of key immune pathways, the homolog and toll pathways, their age-related increase, was dampened in these mutants.
Which fits with the reduced inflammaging idea. But didn't they find something surprising about infection resistance?
They did. You'd think lower baseline immune activation might make them worse at fighting off infection.
Right.
But these Ptth mutants were actually more tolerant to bacterial infection.
Huh. How does that work?
The thinking is maybe their immune response is just better regulated, not constantly simmering with low-level inflammation, but still able to mount an effective, perhaps more controlled defense when actually needed. Less collateral damage from an overactive system over time.
So Ptth is tuning the age-related immune response, specifically in the Oenocytes.
Precisely. And they show this by looking directly at NFEB activation, the movement of Relish into the nucleus in the Oenocytes. It was reduced in the aging Ptth mutants. Same for an antimicrobial peptide. DPTA, its age-related increase, was blocked specifically in the Oenocytes of the mutants.
Okay, the Oenocytes really seem central here. Let's loop back to Ecdysone. We know Ptth controls it via its receptor, torso, in the Prothoracic gland, and feeding Ecdysone reversed the lifespan extension. Did they have other evidence linking Ecdysone, Oenocytes, and lifespan?
Yes, they did another clever experiment. They knocked down the receptor for Ecdysone, but specifically in the Oenocytes.
Ah, blocking the signal reception just in those cells.
Exactly. And what happened? The flies lived longer, and they saw less of that age-related NFD activation, Relish movement, in those Oenocyte cells.
Just like the Ptth mutants.
Just like the Ptth mutants. It really strengthens the case that Ptth influences lifespan through this torso-Ecdysone pathway, acting on the Oenocytes during development.
So it's like a chain reaction. Ptth, torso-Ecdysone, NFP, and Oenocytes developmental timing any lifespan.
That seems to be the core pathway they've identified. But they also wanted a broader view, right? They looked at all the genes being expressed across development.
Right, the transcriptomics. What did that show?
It was a huge undertaking. RNA sequencing across 8 different developmental stages, comparing the Ptth mutants to normal flies. And it revealed, unsurprisingly perhaps, that Ptth influence is massive. We're talking thousands of genes with altered activity levels.
Beyond just Ecdysone and immunity, then?
Oh, definitely. It impacts things like ribosome building, making proteins cuticle development, how the nervous system forms, and critically, energy metabolism, especially during that larva-to-adult transformation.
And did this big data set confirm the role of NF-key during development?
It did. They saw that NF-key-related genes peaked in activity during the larval and early pupal stages, reinforcing that it's not just an adult thing, it's busy during development.
Yeah.
And fitting the picture, the Ptth mutants generally showed lower expression of antimicrobial peptide genes during these stages.
Okay. And they could link Ecdysone to this developmental NF-key activity in the transcriptomics, too.
Yes. Giving Ecdysone back to the mutants partially restored NF-key signaling in larval Oenocytes, and conversely, knocking down the Ecdysone receptor in Oenocytes blocked that developmental NF-key activation. It all ties together nicely.
Okay, so the big picture supports this pathway, but that experiment silencing Relish just in the Oenocytes at specific times feels like the most direct proof, doesn't it?
I agree. That temperature-sensitive system they used was really elegant. It allowed such precise control. And finding that silencing relish in Oenocytes during that early third instar larval stage, that specific window, caused both delayed development and extended adult lifespan, that's incredibly strong evidence.
Yeah, it really pins down that developmental NF key activity in those cells as a key regulator of both processes.
It directly connects the developmental immune state in a specific tissue to the pace of aging later on.
And you mentioned it also reduced the age-related inflammation in the adults.
That's right. Silencing relish early in the Oenocytes seemed to set them up for lower inflammation throughout life. It's like programming for healthier aging.
Did the timing matter? Did silencing it later have the same effect?
Timing was definitely critical. Silencing relish in Oenocytes during the prepupal and early pupal stages also worked extended lifespan. But doing it later, in the late pupal stage, didn't.
Interesting. Maybe the critical window for programming lifespan via this pathway closes by then.
That could be it, or perhaps related to how Oenocytes themselves develop. And importantly, as a control, if they knock down relish everywhere in the body later on, it actually shortened lifespan.
Right, showing it's not just about reducing NFQ overall, but about reducing it in the right place at the right time during development.
Exactly. Context is everything. Tissue specificity, timing, it's crucial.
Okay, so let's try and wrap this up. After digging into all this, what's the big takeaway from this fruit fly research?
I think the main message is that there's this really fascinating, quite intricate connection between how fast an organism developed and how long it ultimately lives as an adult. And in flies, at least, this link is orchestrated by this Ptth hormone signaling pathway, acting through Ecdysone, and crucially involving the NFU immune pathway, specifically within the Oenocytes, those liver-like cells.
And the really mind-bending part is that slowing down development by tweaking this pathway actually leads to a longer, healthier life. It flips the script a bit.
It does. It emphasizes that signals active during growth have these incredibly long shadows, affecting aging decades later, metaphorically speaking, for the fly. Developmental events really do program adult health.
Which naturally makes you wonder, could similar mechanisms be at play in us? In mammals?
That's the million-dollar question, isn't it?
Yeah.
While flies are obviously different, these are fundamental processes: hormone signaling, immune pathways, development, aging. It's plausible that conserved principles are at work.
So the pace of our own early development might subtly influence our own aging trajectory decades down the line.
It's a provocative thought. Could interventions or factors influencing development have these unexpected long-term consequences for lifespan and health span? This research certainly opens up that line of inquiry.
Definitely food for thought. A really fascinating connection between growing up and growing old. Thanks for listening. And as always, don't forget to like the video and subscribe if you haven't already.